everybody knows Chris, so I'll keep this really brief. Chris is off to a job in July at the University of Manitoba in Winnipeg. So moved north out of the US. And he got his PhD at University of Alberta, working with Mark Lewis and Al Wang. And he's been a postdoc here working on a project looking at transients in microbial systems for this is I guess it's 3 years, over 3 years. And he's going to be talking today about some related aspects on modeling and predicting regime shifts in ecological systems.
Thanks everybody for coming. Happy April Fool's Day. You're going to be the fools to listen to a mathematician talking in the CPB seminar. So yeah, hopefully it doesn't scare you away. Hopefully it's like a good DEI type thing. So probably the most important slide, acknowledgements. I want to get them over and done with so I don't forget or miss time later on. So of course I want to thank Alan. Been a terrific mentor through the postdoc. Helped me get a job, of course, which is the goal, right, of a postdoc, I guess. For one of them, I get at least. Of course, my PhD advisor is Mark Lewis and Hao Wang. At U of A, there was a collaborative math biology group. They were a really good group, really a lot of good friends from there. And maybe this is a plug. So Jody Reimer is going to be giving the Math Bio seminar on Monday next week. So I'll send out an announcement, at least to the TPB, but I can send it to CPB if people might be interested in that. So she was a part of that collaborative group as well, and cool stuff. And then, of course, here locally, there's been several research groups that I've been kind of interacting with. So Sebastian Schreiber, Marissa Basket, Caleb Provost, and of course, Alan Hastings. And then, of course, the theoretical population biology group and CPB in general. And then the rest is more or less just where I get money from. So, of course, I have to say thanks to that. I like to get paid. And then also the research, I'm going to talk about some of my PhD work, which was conducted on the Treaty 6 territory. So I kind of have to be really familiar with that. Lived there, recreated there, did some research there. Some of the topics are motivated by a certain lake in that area. And then, of course, we're all familiar with the Papuan people of where we are right now. Okay, so regime shifts, what are they? I'll start by giving some examples of what I would call interesting examples of regime shifts. Starting here, this is the Swedish bowl. And we see these populations has these large oscillations, large amplitude cycles, and then they kind of peter out. They're doing like low level, but persisting, right? So that's kind of an interesting shift from these large amplitude oscillations to kind of this persistent small amplitude. Forage fish is another example. We have this, like, they're not doing so great, and then all of a sudden they can recover and they do pretty good, at least for a couple of years. Right? That's kind of a regime shift that happens, going from this low level, persistent type of population density to an abrupt kind of change. And then another interesting one is the spruce budworm type dynamic. It's a predator-prey type cycle. But if we look at like this part here, for almost 10 years, it looks like we have absolutely nothing happening. The budworm's just kind of chilling, doing really nothing. And then all of a sudden we get an outbreak. And so I call this a regime shift because we're in this regime of nothing's happening, low level, and then all of a sudden we get this huge outbreak, shifting to another regime. And so, yeah. Kind of the main things to take away from this stuff is that these are all kind of not the steady state versus extinction type things that a lot of modelers think about. These are kind of short-term, more ecologically relevant type dynamics that we're looking at what's happening. Yeah, so that's kind of the gist of regime shifts. Now, thinking about more what we can actually do with these things, is kind of classifying them, right? So all these, there's different ways that those regime shifts show up in ecology or in mathematics. And more recently, actually, I guess I got rid of the footnote here, but Alan wrote a paper in, I think, 2018 in Science, or with a bunch of co-authors. They did a really good job of kind of classifying these transient dynamics, and it was kind of a first step into actually being able to really study these things from the modeling perspective. So what I want to do on this kind of slide is just kind of go over just go over how we classify these things, what that means mathematically, and how that's going to relate to actually doing something with ecological dynamics. So the first example is this fast slow dynamic, and that's the same type of thing as that spruce budworm example I just showed you. So what's actually happening is we have two different things operating on different timescales. We have the spruce budworm, typically fairly fast dynamics, especially compared to the tree that's its host. And so the tree will die, and then it'll grow slowly, And while that tree is slowly growing, the spruce budworm can't really do anything. So we get this kind of dissection of two different time scales. And that leads to these types of regime shifts. Another example is these ghost regimes or a ghost attractor. Maybe as a caveat, as a side note, I'm going to use the term attractor and regime almost interchangeably. I don't know, people might fight me on that if they're here, but the idea is that an attractor is like a state where the system's going to go, and then a regime is just like a state that you're hanging out. So if I say attractor, just think regime if you're not familiar with that jargon. Okay, so I call it a ghost regime or a ghost attractor. And the idea here is that if we have this sample of like this trough here, the ball's going to hang out in the trough. That's a stable regime. We're going to hang out there forever. We can't get that ball moving up or down. But in a ghost regime, it looks like a regime in the time series, but when we look at this like slope, it's going to roll really slowly. And so there's the resemblance of something happening there. It's the ghost that's haunting the dynamics, that's letting it stay there for a long time before we tip into a different regime. Right? So here these kind of look the same up until about time equals 100 when we like fall off that ball or fall off that hill. Another example is a crawl by. And so this is a video of what's happening in a crawl by. This is the time series from some model, and we can see that we're staying what seems to be stable. We're staying there for a really, really long time, but eventually we're going to start to head off in a different direction. And so this is still an unstable dynamic, but if you were observing this, say there was some ecological system that had a crawl by type dynamic, it'd take a long time for us to realize that it's actually not stable, right? It's actually going away from it. And so I'm going to talk more about, hopefully at least, talk more about these crawl bys near the end of the talk. And then there's this other idea of kind of pseudo attractors. It's maybe a little bit more abstract, but the idea here is that there's something in the dynamic world that's attracting the dynamics. So we're all going to try to tend to something, stay there for a little bit, and then leave it. And so this is an example of a phytoplankton bloom. And so you can see the difference between phase 1, 2, and 3, and then eventually in phase 4. So phase 2 doesn't really have anything mathematically that's saying this is an attractor, we're going there. But we can see that the dynamics go to that phase 2, stay there for a bit, and then collapse to phase 4. Okay, so now something that's different from these is tipping points. And so if we remember back to January, Karen Abbott came and gave a talk, and she talked a lot about this paper that I was very fortunate to be a co-author on. And so I don't want to spend too much time going through the tipping points because she did a much better job than I ever would be able to do discussing these things. Plus also had a full seminar session, and this is just part of my introduction. But the idea here is that there's something changing in the environment that's causing these regime shifts, whether it be a parameter that's changing, that's completely changing the possible outcomes, whether or not we're adding noise that's shifting between two basins of attraction or two steady states, or there's some rate, so we're changing something at a certain rate, and the rate at which we change it also matters. So tipping points compared to transient dynamics is transient dynamics, they just happen without changing anything. You set up the system, it's fixed, you let the dynamics do what they're going to do, and it's going to be, they can just have a regime shift happen kind of intrinsically to the system. Where with tipping points, we need to actually have some lever that we're pulling and playing with in order to get that regime shift. Okay, so I'm going to throw up this figure a few times throughout the talk, but I'm going to give you a quick rundown of why I throw this figure up here, is that these are 3 different kind of examples of what we might see in ecology. The blue curve is that phytoplankton bloom. The orange curve is just the bacteria population, and the yellow curve is a plant population. And we see all of these 3 things go under what I would call a regime shift. But all 3 of these have a different mechanism that drives that regime shift. So if they all have a different mechanism that drives the regime shift, I can ask the question in ecology, does it matter what the mechanism is that drives this regime shift? And can we distinguish between those mechanisms in order to make like a better prediction? So that's kind of the basis of a lot of things that I've been thinking about in terms of research, is classifying these from just ecological data, not having a model underneath it. And, does it actually really matter to predict these things? I'm hoping that I convince you that it does, but, jury's still out. Okay, so a quick overview of what I want to do. So I just did the overview of regime shifts. That's section 1 done. I'm going to talk about modeling regime shifts. So get some more ideas as to actually what we can do with regime shifts in modeling world, and then how I can predict those regime shifts. And then if there's time, I can talk about some future directions. Okay, so as promised, phytoplankton. I'm going to start there. I think all of us are probably familiar with phytoplankton. The only thing I really want to highlight is that throughout this modeling effort, I just want to highlight that I'm going to assume that really their only growth is dependent on nutrient and light availability. And when I mean light availability, I mean, I'm going to consider that they self-shade, basically. That's like the important factor there. So these pictures I always kind of throw up because they're fun to think about. So this top picture is a beach at Pigeon Lake, which is near Edmonton, Alberta, in Canada. And now I grew up near Pigeon Lake, and I would go swimming there in the summer. My family would take us out, and we'd have a great fun time. And more recently, this lake has been kind of developed. There's a lot of cabins going up. There's a lot more agriculture activity happening there. And so phytoplankton blooms, like harmful phytoplankton blooms, have been becoming more and more frequent. And so this sign, I know you can't read it, but it's like public health advisory. And those signs are actually permanently fixed. And then when the lake is safe to swim in, they just put paper bags or plastic bags over the sign saying, like, it's fine, you don't have to worry about it. And then this is Dry Meat Lake, which is a water source for a nearby town or city called Camrose, Alberta. And I know this looks like it's maybe a pretty blue thing, but you know, you could probably do your best Jesus impression and walk across that because it's all just a mat of bloom. Yeah, it's pretty gross looking, but even though the light doesn't make it happen. And so that's both agricultural reservoir for irrigation and freshwater for a town. So you can imagine, you know, the complications that go into that, having harmful blooms all the time. Okay. And so now when I'm modeling these types of things, phytoplankton, there's two different ways to think about it, or two different kind of frameworks that commonly show up. The first one being the monod growth rate, and the second one being the drupe growth rate. I use this image to kind of distinguish between the two. So the blue cycle, okay, let me take a step back, sorry. Green thing, the cell, algal cell, these little blue dots, the nutrient. And so in the monod form, we just assume that we go from one cell with one nutrient inside of it and 4 nutrients outside of it, skips to this stage here where we have 2 cells and one has that nutrient inside of it. Okay, so this is assuming that the internal nutrient in the monod form is kind of constant. There's a fixed like level of internal nutrient that it needs. And it also assumes that the growth and the division are kind of simultaneous processes. And so that's where we get this S over H plus S. This is kind of like your Holing type 2 functional response looking curve. And so the growth here is depending on the external nutrient. So what's in the medium, what's in the water. The other framework is the drupe growth rate, where we actually divide up the uptake, or we separate uptake and growth into 2 different processes. And so now the growth rate is a function of internal nutrient. So those are the 2 big differences. And so I want to think about the drupe growth rate and kind of ask questions. If we consider that drupe formulation, how does this help us understand transient dynamics and the biological drivers of those transient dynamics? Okay, so scary model. The only thing I want to take out of here is, again, that I want to emphasize it's nutrient and light energy that are contribute to the growth rate of the system. And so that's highlighted in red. Everything else is kind of more or less intuitive, where we have some replenishment of the nutrient. So there's an input of phosphorus and some exchange. So we have rivers flowing in and out. Nutrient is being consumed, so the consumption goes into this function. That's the internal nutrient, it's uptake. So it's all kind of this similar, like we have to eat it, and then we grow, and then we divide and lose some back into the environment. And so back to this kind of dynamic, this model gives us this dynamic. Okay. So what we can do mathematically, if I want to study this model and get some kind of information out of it, we have to do a little bit of work. And so what we do is a non-dimensionalization, or what I've done is a non-dimensionalization. And so in doing a non-dimensionalization, the most important thing to think about is that now F, G, and H are all kind of order 1 parameters. So they're not big, or functions, I should say, not parameters. So they're not big, they're not small, they're kind of all the same order of magnitude. Okay. And so that means that du d tau is like the same order, it's order 1. dw d tau is same order 1. But delta here is a tiny parameter. It comes out when we do this non-dimensionalization with all the parameters that we put in. It's now a tiny parameter. So that means that dv d tau has to be really, really big. We're taking a small number, multiplying it by a big number. So that way we match the order on the right-hand side. Okay, so that means dv d tau is much faster, or change. So v is changing much faster than the rest of the dynamics. And so that's what we get from that's how we get a separation of time scales. So I think, you know, probably all of us have heard of fast slow dynamics at some point. And so if you haven't actually seen the machinery behind fast slow dynamics, this is really kind of a crash course on that idea. Okay, so why do we do that? Well, it's easier because now if I do the separation of time scale, it's easier to actually study these models because I can reduce this to two separate systems of lower dimensions. And the nice thing about it is that there's some mathematical machinery and theorems out there that actually tell us that it's a really good approximation. So that's called exponential theory, if you're like keen to learn more about that. Okay. And so what I'm going to do now is discuss what happens on the fast dynamics and then what happens on the slow dynamics, and then show you how they kind of get glued together in a nice kind of cohesive way. Okay, so fast dynamics, we have kind of two possible outcomes. One case is where the system was nutrient limiting to start, or where there was a lot of nutrient and then eventually the system becomes light limiting. And so, you know, kind of simple looking dynamics, if it's nutrient limiting, okay, let me take one step back, sorry. This is the cell quota on the y-axis. So this is the internal nutrient, and its maximum is 1. So the cell can't take up more than 1 unit of nutrient. So that's why we go, if it's light limiting, we're going to go to the maximum nutrient, and then it's just self-shading itself at that point, right? There's more nutrient in the medium, but it can't do anything more, so it's just going to kind of limit itself by light. And then the other case, it's nutrient limiting, so it can't take up all of it. It's not completely full, but it's kind of going to some level where it's using all of the nutrient that's in the medium. Okay, so then on the slow scale, so the slow dynamics, what's happening on a different time scale, is not going to be the internal nutrient, it's going to be the external nutrient, and it's going to be the actual population, the phytoplankton population. So in the light limiting case, so if we're starting off, okay, yes, I realize I'm getting ahead of myself so much. So I'll go back one slide. So this is the fast dynamics. The fast dynamics should glue to the slow dynamics. So whatever's happening fast is eventually going to turn into the slow dynamics. So if we start off in the nutrient limiting case, in the fast dynamics, when we start in the slow dynamics, we're going to start in the nutrient limiting case. Does that make sense? Yeah. Please stop me if there's any questions. I realize I'm kind of like getting ahead of myself on this, but I feel like I've given this talk so many times and it's like an autopilot, but I'm forgetting what I actually am supposed to talk about. Okay, so yeah. So these are supposed to glue together. Fast, slow dynamics, they link up in the middle. So fast dynamics, we have nutrient limiting or light limiting case. Same thing in the slow dynamics where they link up. So in the light limiting case, we see we kind of have the start of the bloom that's happening, and then the nutrients are just being depleted. And we see we have a negative situation here. So that means at some point it goes from positive to negative. So eventually that's going to have to transition from being light limiting to nutrient limiting. And if that's the case, then we just have the nutrient limiting case where everything kind of just goes down to 0 or to a low level. Okay, so hopefully now this all makes sense when I put you in this final picture. And I think, was it you, Jenny, that was saying like pink needs to be in more figures? So shout out to Jenny for more pink in figures. So we're starting off our initial conditions. So the initial point, how polluted is the lake kind of thing, is these red circles. And the fast dynamics are these doubled arrows. And we see we go from the initial condition, we go as fast as we can to the light limiting case. And then we're on this light limiting kind of sub-attractor, pseudo-attractor, and we start to head down. Right? This is what the dynamics is telling us to do. And eventually we transition to the nutrient limiting manifold. And then we go down to the blooms over. And if you kind of tilt your head sideways, you can see that this actually represents that bloom, where we go like this and down and then over, right? If we like rotated it up, it looks the same. And so this is really kind of a nice result because it tells us like the dissection of this manifold very clearly is light limiting when you look at the equations. This manifold is very clearly nutrient limiting when you look at the equations. And so the bloom is really just governed by how long does it take to switch from one manifold to the other. And when we kind of realize that, we can write these kind of approximations. And I say approximations because if we write the whole formula out, it's a little bit complicated and it's a transcendental equation, so you can't actually solve for the variable you want, blah, blah, blah. So we do these approximations to say how long is the bloom going to last based on the initial kind of total phosphorus. So if we have like a lot of runoff in the spring in a frozen lake, how much nutrient was input, how long will that bloom last based on that input? Okay, so let's take a water. So the one thing I didn't consider yet is that this is a phytoplankton bloom, and I just talked about like spring runoff and nutrient pollution. And so one obvious factor that drives that is human interaction. And so I'm not the first person to think about this. This paper, this figure comes from a paper from Chris Bao and Maduro Nod's group, if people are familiar with them from University of Waterloo. And they really just, their whole kind of research group focuses on highlighting the connection between an environmental system or an ecological system and the human system. And they come up with a lot of ideas, and it's really interesting work. And so that's kind of the next step for this model, or this kind of regime shift framework, is thinking about how is the system influenced when I actually explicitly consider a human dimension. And also other people think about it, and this is an example from Sustainable Development Goals of the UN, where they think about this wedding cake, biosphere, society, and economy kind of all nested together in terms of this, reaching the Sustainable Development. Okay, so now how do I actually do this? I'm going to think about it from a game theoretic type approach, where I have a cooperator and a defector. So cooperator being the group of people that would pollute less at a lake. Defector are the ones that either keep the status quo or pollute more. F here is the proportion at a lake. So I have a single lake, and I'm just normalizing it so that there's a proportion of cooperators, a proportion of defectors. And the interesting thing to think about is what I want to model here is how does a defector perceive their cost, and how does a cooperator perceive their cost? So I have a few things. I say that they each have a base cost just to cooperate. So if you're a defector or a cooperator, there's some cost associated with that. For example, a defector would have a lower cost because they're probably not paying for like a septic tank or sewage treatment or something like that at a remote cabin. Where a cooperator probably has higher cost because they're putting in some money or some effort to reduce their pollution. Both of these both the cooperator and defector have a cost of a bloom associated with it. You can think about that as if you own a cabin on a lake that has a bloom, you're both facing the same like negative recreational costs or property value cost associated with that bloom. And now the defector has an additional cost based on social pressure based on the bloom. And I can think about that as like social ostracism. So you have to assume that the defector here has somewhat rational thinking, knowing that the bloom is bad and they're polluting, meaning it's bad, and that their neighbor, if they're know it's bad, the whole community says it's bad, you should feel bad. That's ostracism. So that's a social pressure, which is a little bit different than a social norm cost, where a social norm cost is saying, well, what is everybody else doing? Should I switch? So you can have a social norm cost just doing what people are doing, even though it might not be the right thing to do, but it still could be a low or high cost depending on the situation. I used to use the social norm cost as an example in Alberta of like electric cars versus like fossil fuel cars. But I think that like, buying a Tesla is now nuanced, and so I'm just going to skip that. So, okay, so when I consider these types of dynamics with a human population, basically this is the penultimate result is, this is the main result is different types of regimes that come out of a human influenced environmental system. And so the COOP here is short for cooperation. So we have a mixed level of cooperation, low level of cooperation, high level of cooperation. And on each of these different axes, I'm just thinking about a different parameter. So this would be like a social norm cost of defecting, social norm cost of cooperating, based on like how many people are doing it. Okay, so mixed cooperation here means I'm now thinking about a bunch of lakes in a network that are socially connected, not ecologically connected, but socially connected. And so if they all kind of start off on a mixed kind of regime, each of them, so some are cooperating, some are defecting, and we're in this parameter region, and we don't expect anything to change, right? Everything's just kind of going to stay as it was. But if we start to increase, like say, social norm cost to defect, then we can move to this high cooperation regime. Or if we decrease something and we're up here, maybe we'll move to the low cooperation regime. So this is what we would call like a 2-parameter bifurcation plot for the math people in the audience. And so we can do this for any number of parameters to get an idea of like, how do we pull certain levers to get regime shifts or to avoid regime shifts. And then the next layer of this is what Karen talked about was rate-induced tipping. So if we're actually starting to think about these campaigns and how you would implement them, most of the time, if you're thinking about subsidies or fines or something like that, they're maybe increased over time. Sometimes they're abrupt, which is fine. If they're abrupt, then that's what we would get on the previous slide. But if they're increased over time or decreased over time, we can still get similar favorable or unfavorable outcomes, but maybe at a less like magnitude, right? So we can be kind of careful how we actually do this. And so in this figure, we are removing some sort of anti-pollution campaign, so we're removing some sort of subsidy or we're adding fines or something like that. And so we're starting off in the blue curve for no rate, nothing's changing. So we go to this high level of cooperation. But as we start to increase that, that anti-pollution or decrease that anti-pollution campaign, we actually start achieving either that maximum cooperation later on, or we completely tip and we have a low cooperation outcome. So then the opposite can occur if we're implementing something, adding something. So before we were going to an unfavorable regime, and now we're adding it, we're going to a favorable regime. And it depends on the rate that we're doing. Okay, so this is, nothing's changing in the system other than that single parameter, and it really depends on how fast we change it, right? So even if I changed it instantly, we would still be in the same regime. So it's interesting to change it, how the rate of it. And so this is another example that I've kind of, this should be more future work, but this is an example where we have periodic changes in these parameters. So they're slowly oscillating over time, and depending on how big the amplitude of those oscillations are, we get a similar type dynamic. If the amplitude is really, really big, we get this type of tipping that's happening. And if you're keen, you can notice that we actually get a really interesting result where we tip, and then, so we don't tip, we go back to tipping, and then for really big amplitudes, we actually don't tip again. So that's something that we're trying to explore. It's kind of a fascinating result. Okay, so kind of summary of the first part there. So yeah, I was kind of trying to show you that we have these mathematical tools and these mathematical kind of frameworks that can help some, I made a pun, I guess, shed some light on biological mechanisms that explain these transient phenomena, these regime shifts. So we gave these interpretations of how these regimes can happen, or regime shifts can happen, and we also kind of want to emphasize that there's more than one route often to get these regime shifts that happen. Okay, so now moving on to predicting regime shifts. So there's been lots of work done, especially more recently, kind of in my generation of science at least, to predict these tipping points or early warning signs for tipping points. And this is a review paper done in 2024, so fairly new paper by Vasilis Dakos and his group, just summarizing the different types of regime shift or early warning signals that are out there. And I guess you probably can't read it, but this is variance, autocorrelation, skewness, power spectrum. I'm not going to go through them all, but the most popular ones are variance and autocorrelation for sure. And those are from what we would call critical slowing down theory. But the idea here is that there's some sort of statistical measure based on a time series that will indicate a tipping point. So even though we have all these different ones, and this is a very important kind of question to answer, is how can we predict these tipping points? We have early warning signals. Most of them are based on some sort of statistical measure of the time series. And so what do I actually mean by some statistical measure? I'm going to talk through the example of autocorrelation here. And so this is used to predict the bifurcation. So it is predict the tipping point, right? So there's some bifurcation that's happening. So this is a time series that undergoes a bifurcation. So slowly we're changing some parameter over time. Eventually we change it too much that we go into a different regime. And on these plots, this is just x and x t plus 1. So these are kind of autocorrelation plots. So you can see when we're far away from the tipping point, there's really no autocorrelation. As we get closer and closer to the tipping point, we can see the autocorrelation starting to increase, which is this curve here. So autocorrelation itself is a statistical measure. Is it starting to increase or has a positive trend? Then we can kind of use that as an indicator that we might be getting close to a tipping point. Another kind of more popular method is using deep learning or machine learning or AI to predict these things. So here, this is an example from Tom Bray. So this is back from the Maduro group and Chris Bow's group, where they're taking a time series or a made-up time series and using machine learning to predict the probability or to give a probability of a tipping point happening. And these are 3 different plots of 3 different types of tipping points, or I should say 3 different types of bifurcations that are going to happen. And so it does a pretty good job. It's a really kind of useful tool, perhaps on an idealized time series. Okay, so kind of what is missing though? The main thing is that what I've talked about are these statistical measures that are predicting a tipping point. What about a transient dynamic, right? Because all those tipping points, there's some parameter changing, something's changing in the environment or demographically. But I want to ask the question, what's happening if it's just an intrinsic tipping point or regime shift in the system? Did those types of early warning signals actually work? Theory would suggest that there's no reason that they should, but there's also no reason that they shouldn't. We kind of just want to explore this. What can we do? Is there more machinery out there that we can look at? And so, what can we do? I don't need to convince anybody here that ecology is complex, right? But, I'm going to do it anyways. So these are examples of a marine food web. This is an example of anaerobic digester, which is what I've been working on with Alan since I've been here. And if I was to actually write a model for this whole system, this complex system, like that's a huge endeavor, right? Each box would be one equation and the arrows might be multiple. So yeah, it's just way too much. So, for a math biologist, I can only do so much. But there is this kind of new idea of empirical dynamical modeling that I'm going to get into that we can maybe actually say something about it. So as a crash course, this is kind of what we currently do when we're doing modeling. We have some observable variable, x of t. We write some model. So x of t plus 1 is a function of the previous time plus some noise. And when I say noise here, I don't just mean like it's like a white noise. It's just you add some stuff to it. It could be a temperature dependence or whatever, but it's some function of some noise, some added stuff to make it make sense. Okay, so yeah, that works. That's what we kind of all do, but it's not quite precise, right? We could be a little bit more bang on with it, saying that if that's an observable variable, we also have unobserved variables. So y of t now can be my unobserved variable. And so really, x of t plus 1 is a function of all of the observed variables and the unobserved variables. And then of course, y of t is a function of the observed and unobserved variables. Right, so that's kind of more really what's happening in ecology. So how can we kind of embrace these unobserved state variables, right? It's a huge question. And so this is where Tauken's theorem, a really important theorem in mathematics, come from. And so since it's so important, I'm going to take a second to go over the proof of it. That's my April Fool's joke. I actually don't even know what the proof looks like. So the theorem is just expressed in this video, which is much more palatable. So in the left here, these are kind of the original coordinates, x, z, and y, and x, z, and y represent a producer, grazer, predator. So just a competing or a tritrophic system doing its thing. And so on the right of this, we have delayed coordinates. So x of t minus tau, which is just some time step, and x of t minus 2 tau, which is another time step. And what we can see that's happening in this video is that even though they don't look the same, the black curve and the red curve, there is a one-to-one correspondence. Like this, they're moving together. And when the black dynamics slow down, so do the red dynamics, they slow down as well, right? So what this is saying is that, okay, I can kind of recreate some type of dynamic just looking at the right-hand side with just one variable, right? I don't need all the other three variables because they're the same. So there's a map from this delayed coordinate system to the original coordinate system. And that's kind of really the key of like Tauken's theorem and empirical dynamical modeling. So what I'm really saying is that using this framework, we can reconstruct system dynamics like the pseudo attractors, like transient dynamics, and just from looking at a single time series of one observation, right? We can map this back to this somehow, hopefully. Okay. That's kind of the gist of it. That's the goal of it. Okay, so in practice, how does this actually work? Okay, I'm going to skip this part because it's really not, it's interesting, but it's kind of not that important. So in practice, what we would do is just make a linear map from one data set to another using all these tables. And so the question here is, can we predict transient dynamics from ecological data? And so this is where the idea of anaerobic digestion comes in. So anaerobic digestion, for those that aren't aware, is this idea where we take simple substrates, use some bacteria to convert the substrates into volatile fatty acids, and then use another type of bacteria to convert them into biogas. So super simplified type of model here or assumptions, but that's kind of the gist of it. And so the biogas is used for power sources and water or sewage treatment and some other useful things. And so from that type of model, what I can do is create this synthetic data that has this type of transient dynamic where it's kind of going to some pseudo attractor and then it's falling out and ending at a low level kind of persistent bacteria state or density. And so these dots here are what I'm going to say representing the transient dynamic end time. So we have a transient dynamic that takes a bit of time and then it ends after some time. And I do this all deterministically. So this is all of the math underneath the hood, right? So I write a model and I say, okay, I know how this model acts and I know the transient dynamics of the model. And so I use that model to make those dots based on this time series. Okay, so I know where the transient dynamics end. That's great. And I did it all through math. And so I'm using that as a starting point to think about if I'm going to develop a new method for early warning signals, I have to compare it to something. So I have to kind of come back to that eventually. Yeah, I just said that. Okay. And so using this SMAP forecast or the empirical dynamical modeling forecast, so I'm reconstructing the attractor from the data, and then I'm making a forecast from that, we can come up with these error curves, right? And the one thing that we notice is that originally the error curves stay pretty low, and then they have some weird spikes in the middle, and then they go back to being low. And that makes sense from the empirical dynamical modeling literature, is that if we're on an attractor or near an attractor, that means there's a lot of data there. There's a lot of density in that, like the red versus black curves. There's like a lot of density there. So our prediction should be quite good when we're on an attractor or near an attractor. But when we're moving between attractors, there's no density of data. So our error should be pretty high. And so that's what corresponding to these spikes, right? So we're starting with low error, and then we have a spike, meaning we're transitioning from one attractor to another. We get these spikes, and then we come back to it. So I can use those spikes as kind of a different type of early warning signal, saying that we're leaving the transient soon. Yeah, so I want to predict those big dots that I gave you on that first pseudo example. And so then the results look something like this, where I'm using R squared to compare the predicted from my new early warning signal to the deterministic setting. So here, if it's yellow, that's close to 1, so that's doing a really good job in R squared. And it is just against the regression line y equals x. So I want to make sure, like, if they're equal, then I'm thinking it's doing a good job. And so, yeah, there's some nuanced ideas about like what's happening here. This isn't so good. And that has to do with the framework of the empirical dynamical modeling literature, where if the time points are too spaced or not spaced enough, then we're actually like overcompensating and we're kind of making up an attractor that's not actually there. Right? If it's like too condensed. Too condensed and too many data points, I guess, is the difference. And so, yeah, so this method actually does a pretty good job, right? There's quite a few regions where there's yellow. If I'm increasing the time step of the data, we get a little bit worse kind of predictions, but it still does a decent job. If we're increasing or decreasing the number of sample points, so this is what kind of the data would look like. Yeah, so then on each individual plot, the x-axis would be the error threshold. So that's like the spike. How high does that spike need to get before I actually say that that's the transient endpoint? And then this is just different noise as we're going up. So if the system's really noisy, it makes sense that it's not going to do so good because it's just dominated by noise, right? It's more of a random system than an actual deterministic system. So yeah, great. So that does a decent job. I'm happy with this result. And this was kind of the first paper we published since I've been a postdoc. Okay, so now something that we're working on. Hopefully we have a manuscript out soon, but the idea here is with the crawl by. It's a different type of dynamic. It's not this like attractor to attractor. There's a single attractor, sort of. It's this stable equilibrium or unstable equilibrium where we go to it in some direction and we repel from it in another direction. So you can say this blue line is attracting, this yellow line is repelling. And it must have a strong attraction in some direction and a weak repulsion in another direction. So that gives us this crawl by type dynamic. And so I'm going to reiterate that when we have this kind of dynamic that's going here, if we're trying to make a prediction about how long is this transient going to last, it's really hard to know, right? Until we kind of start to get away from it. So this is an interesting question or interesting problem as a crawl by dynamics. And the other kind of more generalized thing about a crawl by is that most of these types of transient dynamics that we've looked at can actually be thought about as a crawl. This is maybe a bit abstract, but can actually be kind of thought about as a crawl by with like maybe one less variable or one more variable. Like, so in a different type of dimension, everything eventually kind of collapses to a crawl by more or less. So it's worth thinking about how do we actually predict like what's happening with a crawl by type dynamic. And so I just quickly giving like a rundown of the preliminary result. On the top, we'd have the time series. So this is some made up noisy time series based on just a linear system that has this saddle type behavior. These are the second one is the established early warning signal. So this is what people have been doing for a long time. I'm using the covariance and the autocorrelation. And then the bottom two are these like new measures. This one is, I say newish because that's from the first paper with the error. And this one is just these EDM measures about predicting the direction that the system should move in and also predicting the magnitude of the next time step. And so we can kind of see if we zoom in that the variance starts to work maybe right about here, where it could act as an early warning signal. We see it kind of like dips down and starts to come back up. So maybe the low point would be where the variance starts to increase is usually the signal. So it might, it starts to increase here, but we might not be able to get the signal until maybe a couple time steps after. So it starts to work, but I think also the eyeball method beats it at that point, right? We can just look and say, yeah, it's increasing. It's not a stable time series at that point. And the same argument can be made for these autocorrelation, right? Where it's starting to increase and it's like kind of capping out. So those kind of established early warning signals for this example don't really work in terms of predicting where there's no trends that are obvious or more obvious than the actual time series itself. But now just looking at this, like, so the direction here, it starts to kind of stabilize. Maybe you could argue it stabilizes here, but that's pushing it. It starts to stabilize here. And that kind of corresponds to it's attracted to that weak repelling eigenvector, or that weak repelling direction away from the equilibrium point. And then same with these old ones, we have this like spike initially. I can't really explain that one that well. This is again like preliminary results that we're going through. But we can see that the magnitude or the absolute error again, like does this increase that we would expect it to based on this like type theory. Okay, so yeah. And then comparing it to a stable node. So this is what we really want to compare it to is it going to be a transient dynamic type happening, or is it going to be like a persistent type thing happening, like a stable dynamic or stable regime? And so comparing these two things, that certainly these two, like they're noticeably different, like at this point, in this point. And perhaps the old early warning signals could give a lot of like false positives. Okay, so kind of maybe ground us back to the original question or the original idea is distinguishing between these mechanisms of ecology and whether or not it actually matters. So the established early warning signals that have been around for a while now really focused on predicting tipping points due to some change in some parameter, so some bifurcation predictions. And I think I've hoped to at least explain to you that if it's a transient dynamic, those early warning signal signs don't really do as good of a job as we would hope that they do. And so that there is also the possibility of some new metrics based on this empirical dynamical modeling literature that could help with that. And so kind of a quick recap. So I discussed the various ways that regime shifts occur in ecological time series. That was kind of the first part. And then I went on to establish some new tools that would help predict these regime shifts or help predict the difference between a crawl by or a stable kind of system. And so, yeah, hopefully at least being able to use these types of tools to hint towards the underlying mechanism can help us, make better predictions in the future towards transient dynamics. Okay, so now kind of moving on to some future work quick. I think I have some. So yeah, the next idea is to use this empirical dynamical modeling or EDM framework to think about how we deal with missing data or like hard to get data. So in a lot of systems we have out there, there's especially what we're working with the anaerobic digesters, it's really easy to collect things like pH, for example, right? It's easy to collect pH, we all know how to collect pH, but it's really hard, or it's really expensive at least, to get like the gene sequence of that bacteria, right? That's a lot more involved, a lot more expensive. And so using this type of delayed coordinate, I'm curious if we actually need to get that expensive data, right? There might be enough variables out there in that system that are cheap and easy to get, and if we collect them in enough abundance or regularly enough, we might be able to still be able to say something about the system. And so the main idea for this comes from the idea of causation. So I think we've all kind of maybe heard of at least Granger causation or causality. And the idea there is that some information about one of our variables is hidden in another variable, right? So when we combine both of those together to make some model, we get a better outcome than if we were just to have one alone. So the same kind of idea applies here in this EDM framework, just a little bit kind of twisted upside down. So again, here in this top, we have this blue circle, and this is an area of our like attractor or dynamics. And if this blue circle starts to become more and more dense with data, then we would assume that both the green and the red circles, which are these like shadow manifolds, so this is only the Y, like this is only using the Y time series, and this is only using the X time series. So as we increase the density in that blue circle, we should also be increasing the density of data in the green and the red circles. And so we can use that idea with EDM to say, well, okay, if I'm throwing in a bunch of data here in green, so I have a bunch of data for Y, like it's useful data, I can just make this more and more dense, and I know what the dynamics do in this Y kind of shadow manifold, I can say something about what's happening with X, right? Since there is a map from here to here. And so that's the idea of using that kind of, causality, I guess, so to speak. It's called convergent cross-mapping, but using that idea to say, well, we maybe don't need that data. We can use what we have in this variable alone, just embed it more, try to get more data of it, and try to map that to something that we're actually kind of curious about. And so I've done some kind of more preliminary work again on this. To these curves, you're just saying this is the error prediction or prediction error based on these different types of embeddings. And of course, they're different, right? And some are better than others. So, you know, like just using U and X, they don't really do so good. But as soon as we throw in V, the yellow curve does a lot better. Right? So this is just an idea that this is a promising kind of maybe outcome. And then the other idea of future kind of direction that I'm going to focus on a little bit when I get to my new spot is predicting these types of ecological changes. So I did all this machinery using my math background and whatnot, but you know, it doesn't really matter until it's useful, right? It's a good idea to do it, but in reality, we want to actually be able to predict these types of things. So this is a time series from a collaborator that's at the University of Tennessee. His name is Richard Zhao. He spends so much energy getting these time series. So this is anaerobic digester, and he's modeling. I just show the time series of pH. And we're doing these perturbations where we change the temperature of different replicates. So the purple is changing it to 44 degrees Celsius, the yellow is changing it to 41, 38, and then the control is at 35. And so when we add this perturbation for a little bit of time, so just over 50 days, you know, everything starts to change, the composition is starting to change, the pH is starting to dip, and we're worried that the reactors are going to collapse, right? And then we take away that pulse, we put everything back to 35 degrees, and then we get this recovery, slow recovery for some, but we still get a recovery. But the interesting thing is that this purple curve actually kept decreasing for quite a while before it started to come back. And so, you know, can we do anything with this? Like, can we predict this ecological change? If we were monitoring this and we took it off and we said, okay, we're screwed because this is still decreasing, it's collapsed. We might do drastic measures to try to like get it from stopping to collapse, but they might be unnecessary in this situation, right? We could have just let it recover after some time. So in terms of monitoring and actual like, you know, predictions of these types of things, I'm really interested in using this type of framework and this type of regime shift in data from data to actually, you know, try to predict something. Okay. And so next steps, I'm moving on to, as Alan mentioned, I'm moving on to the University of Manitoba. So, you know, I guess if anybody's interested in staying in touch, and curious about more transient dynamic stuff, you can look for my webpage at the University of Manitoba. I'm also going to be, you know, starting to build a lab in the next couple of years. So if you have any like interested undergrad students in your lab or master students or even postdocs at some point, hopefully, please do reach out. I'm excited for the next steps. And with that, I'll take any questions.
Okay, it might be a silly question, I don't know. So do fast, slow, transient dynamics, if you looked at a time series, could you tell that's what it was? Like, is it something that is identifiable?
Yeah, so if you have data on the thing that is moving fast, I think it would be more identifiable. So the example would be like that spruce bloodworm, where it's like doing nothing, it's staying there for a long time, and the outbreak is like really quick. Where if you were looking at the tree time series, it's a lot more like doing this. Right? And so that way you might just think it's some cyclic thing, like normal periodic dynamics. So if you're able to capture the dynamics of the fast thing with how you're collecting data, then it's a lot easier to distinguish between the two.
You see the signal of the slow thing in the fast.
Yeah, so there's certain things because I mean I'm saying it's nice and smooth, but it really isn't. I wonder if I actually have all the way back. Oh, now it's okay, I can't go back. If you remember in the one picture, I had the fast slow where I had like really big arrow and a small arrow, and it kind of made this like duckbill shape. So if you're looking at, if you can construct that type of attractor, then you can see the slow dynamics would have that like cycle. I wish I had the picture. I'm doing it that helps. Yeah, okay. Yeah, no, it's a great picture. Yeah, I don't know. Does that answer your question at all?
Yeah, yeah, thanks.
Early on, when you were talking about the phytoplankton, you made the assumption that it was either limited by nutrients or limited by light. And I guess I've become skeptical of the idea that there are single limiting factors. If you have interacting limitation, co-limitation, or if light and nutrients interact, can your framework deal with that kind of situation?
Yeah, so that's a great point. Like, there's definitely the co-limitation that's going to happen here. The framework that I've been, or the idea that's based on is like the stoichiometry, which is kind of like Liebig's law of the minimum. So it's just a minimum function of one of the things of whatever the things you put in there. And so, you know, in mathematically, when you multiply the two together, it's not that different than just taking a min function. So that's what we did there. So the framework certainly is like capable of thinking about co-limitation with these interactive effects. What would happen is the model would have a different function form there. People have certainly done this. I don't know exactly what the function would look like. I mean, I suppose like just the product of nitrogen growth rate versus phosphorus growth rate might be enough of an interactive effect to get something. But I do think that as the system becomes like less eutrophic, that's really what's driving that dynamic. So you're starting off in like highly polluted kind of regime, and the dynamic or the nutrients are slowly being depleted through growth or through like output through a river or something like that. So I think that type of dynamic would probably still stand true, at least in the model. I'm not exactly sure how that would look though, in terms of like how different the dynamics would look. Go ahead.
So I have a general question about the embedding dimension approach that I was thinking about when you were talking about it. It's obvious to me that it works if the system is closed. But,
Yeah.
you know, if you have a little bit of input from outside the system, like you can think about a predator-prey model and you can rewrite it as one is the lagged function of the other. But what if one of them, say the predator, has a little bit of, you know, birth rate that comes from
Yeah.
something other than the
Yeah.
prey that's outside the system? Have people looked at how robust that general approach that's suddenly become trendy is?
Yeah, so
to-
yeah, it's one of these things so that like going back to basically Tocquen's theorem, I know I didn't go through it, but the idea is that there exists some embedding dimension, so the number of embeddings where you can recreate the native attractor. So assuming if you're having this really complex predator-prey system, the number of dimensions of that system could be 100, right? And then if you're embedding it, if you just go to 150 embeddings, then it's going to do it. So theoretically, yes, it's going to cover all of that. In practice, what we usually do is just do enough embedding dimensions until it works, which is a very unsatisfactory answer. I totally get it. But hey, it works. So like, why? I don't care. All right.
This makes me want to go home and play with the algebra, and I feel like if you just add a little bit of exogenous something to the predator, you can't do the algebra anymore. But
Yeah.
anyway.
So the one thing I will mention too, and I'm sad that I can't put the figures back up, but when I gave you that, when I did that prediction, it was one time series, and I was only doing the prediction on that one time series, but the model was three-dimensional, right? And so I only had like one or two embeddings. So in terms of kind of pretending, like basically what I've assumed that there's another variable that's doing some stuff that I'm just pretending it doesn't or ignoring or don't know it exists. So in terms of like something else happening, more variables working on, you can kind of get away with ignoring them, and it still works. Of course, what I did here was an idealized system. I made my own data up, blah, 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 but it's a proof of concept at least.
Cool. Thanks.
You made like this passing comment about like everything is a saddle bifurcation or
Yeah, saddle crawl.
yeah. I don't know. Can you say anything more on that?
Yeah, well, I can try, I guess. It's like I said, a very abstracted idea. Like you can think about if you have, I guess go back to the phytoplankton dynamic where I have this like attraction to some thing, right? It goes up and then it stays there for a while and then it leaves. So you can think about that attraction to it is kind of the same thing as that strong attraction versus the weak repulsion, right? And so now if I was to model that in a higher dimensional system, that there's some kind of other mathematical framework where basically that attractor is now a higher dimension, where it has an attracting and a repelling eigenvalue. And then all of a sudden it is just a saddle, but in like 6 dimensions or something. So to have a saddle, all that you need is one repelling eigenvector.
That's like coming from like this unobserved, like some unobserved dynamics, like potentially one of those, like.
Yeah, kind of more or less, yeah. I guess that's what yeah, that was probably,
Yeah.
it's a bit much.
All right, well, let's thank Chris for his talk.